Okay, so this part of the lecture is going to be about the stimulus for contraction and the actual contraction itself. So hopefully you've done a reading and remember that the contraction happens because of a motor neuron. So a motor neuron is going to send some signal from the brain through the spinal cord, or even directly from the spinal cord, some signal down the motor neuron, switch to green, and that signal is going to arrive at the muscle. Let's talk about what that looks like. So this is the motor neuron, and the end of the motor neuron is the synaptic knob. So it's a synaptic knob. And then where the nerve and the muscle meet, there's a small space in there. That small space is called the synaptic cleft. So as the signal arrives, remember it's an electrochemical signal. It's coming down there. That signal, when it arrives here, causes calcium's +2, to flow into the synaptic knob, and it causes these vesicles to fuse with the end of the synaptic knob. When those vesicles fuse, they release acetylcholine into the synaptic cleft. And that acetylcholine binds the receptors on the motor end plate, which is that folded part of the sarcolemma, and that sends a signal into the muscle. So go through this. The signal comes down, reaches the synaptic knob. That causes calcium to flow into the synaptic knob. The presence of the calcium in here causes vesicles containing neurotransmitters to fuse with the motor end plate, or to fuse with the end of the synaptic knob. They release the synaptic the synaptic vesicles release the neurotransmitter into the synaptic cleft. That flows over the synaptic cleft and binds to receptors on the motor end plate. Okay, and we'll pick it up from there when we get to the muscle contraction part. And then the other part of this is the sliding filament theory really just shows how those myosin heads pull the actins. And when the myosin head pulls the actin, it shortens the sarcomere. So really, those H zones and I bands disappear or get much smaller because the myosin heads are pulling on those. This leaves you a lot of room because there should be 2 pages in your PowerPoint to write notes on, but you may need a little bit more. So a couple of things we need to talk about before we do any of this. Well, first of all, this is on page 297 to 301 in the 14th edition of your textbook, and a summary of it is on table 9.2. A couple of things that we need to talk about first is that ATP, and you remember this from the unit we did earlier about chemistry of life, ATP is the high energy molecule we use to power our cells. When we need energy, ATP is broken down into ADP and a phosphate group, okay, in inorganic phosphate. So this is the high energy. ATP is the high energy one. ADP is the low energy one. All right, so first step that happens is that we're actually going to start with the action potential, or the nerve signal coming down the motor neuron. All right, nerve signal comes down that motor neuron. I'm abbreviating because this thing does not write very well. When it reaches the synaptic knob, it causes calcium to rush into synaptic knob. Okay. Presence of calcium causes synaptic vesicles. So presence of calcium causes synaptic vesicles to fuse with the membrane of the synaptic knob. When the vesicles fuse, they release a neurotransmitter called acetylcholine. ACETYL choline, CHOLINE, to flow into the synaptic cleft. Okay. The acetylcholine, which is abbreviated capital A, capital C, lowercase h, binds to receptors. What's going on with this thing? Binds to receptors on the motor end plate. Okay, so it binds the receptors on the motor end plate. That opens up sodium channels. Okay, and sodium flows into the muscles. Okay, so sodium flows into muscles, and that's an action potential. That's what an action potential is. Okay, and that's equals an action potential. So the action potential is just that sodium flowing into the muscle, and it travels down the muscle in both directions, and it also travels down the T tubules. So action potential. Travels down muscle. Okay, this thing is so choppy. Down muscle. And down the T tubules. When they travel down the T tubules, it causes release of calcium release from the sarcoplasmic reticulum. Oops. Okay. I'm actually going to just type the rest of that. All right. So I forgot what number we're on. 6 or 7. Let's just say 7. So calcium is released from the terminal cisternae of the sarcoplasmic reticulum. That calcium is going to bind to the TNC of troponin. And remember that TNC always has 2 calcium bound to it, but it needs 2 more to move. And then that causes troponin to move tropomyosin away from the myosin head binding sites on G-actin. Okay, so the troponin is going to pull the tropomyosin away from those binding sites on G-actin. As soon as the myosin head binding sites are available, the myosin heads bind to G-actin. When that happens, the ADP and the phosphate group leave the myosin head. Okay. As soon as the ADP and the phosphate group are gone, the myosin head pulls G-actin, or pulls actin. And that's the contraction. Okay. Once it's pulled it, it's stuck there. It can't move. And then it won't move until a new ATP attaches to myosin head, and that causes the myosin head to be released from G-actin. Okay. When it's released, the tropomyosin can cover those G-actins again. And now the ATP breaks down into ADP in the phosphate group, and the myosin head returns to the high energy position. And basically, the high energy position is the myosin head is cocked. It's ready to contract so that it can. So as soon as those myosin head binding sites are available, it can attach. Okay. Relaxation happens when acetylcholine esterase breaks down acetylcholine in the synaptic cleft. Otherwise, you're just going to keep getting a muscle contraction. And sometimes there are some bacterial toxins that can be produced that stop that from happening, stop you from being able to relax that muscle. So, for example, tetanus, that toxin can cause that. Can cause the acetylcholine esterase not to be able to do its work. Okay, muscle impulse stops, calcium goes back in the sarcoplasmic reticulum, and the myosin and actin binding is prevented because the tropomyosin is in the way. Okay, next part we'll pick up here. So if you can, go to Get Body Smart before that.